So, Jamie, are you ready?
I hope so.
Okay.
The telecom system has gone down today, so there's been no Wi-Fi. So
I
my
see.
colleague Ikos, I'm tethered to his phone, so hopefully this is going to work.
Okay, yeah. Well, okay, now we will continue with the presentation from Dr. Jamie Craggs about coral spawning in action, harnessing ex situ and in situ techniques for research, species conservation, and restoration. And Dr. Craggs will be able to discuss with us after his presentation, so you can leave your questions in the chat box during or after his presentation. So, Jamie, the time is yours.
Thank you very much. I will share screen first and let's... Can you see this screen in presentation mode?
Yes.
Yes.
All good.
Wonderful, wonderful. Thank you. Thank you for asking me to speak. I'm going to talk about some work that I've been doing for the last 15 years, looking at understanding coral reproduction, ex situ, so work that I've been engaged with in London for the last 15 years, how we're using those techniques to better understand coral reproduction, for species conservation as well, and now that I work for Mars Sustainable Solutions as well, how we're intending on applying these sexual reproduction techniques for in situ conservation. So just to sort of introduce you to myself, what kind of ties all my work together is coral reproduction. I currently work...
Kerja sekarang tentang reproduksi karang.
Jobs through employment. I work in London at a museum called the Horniman Museum and Gardens, and I'm the principal aquarium curator at that museum. And my work there focuses very much on how do we reproduce corals in aquarium environments? How could we use those aquarium environments to better understand coral reproduction and using that for species conservation? So I now work, I've been at the museum for about 18 years, and in the last year I've reduced my hours, so I do about 40% time now at the museum, and that's allowed me to join Mars Sustainable Solution team to bring some of that knowledge of coral reproduction and support the restoration that the Indonesian team are doing. And then another area of my work is I set up a business a few years ago called Coral Spawning International, and we work in partnership with a company in the UK where we build coral spawning systems and send those around the world to support different groups, both coral reproduction research and their reef restoration efforts. So using land-based spawning to generate genetically diverse corals and using that to plant out to coral reefs. So I have a bit of a portfolio of work these days, but very much focused all around coral sexual reproduction. So as Trias highlighted, unfortunately this is a common sight around many parts around the world, whether that be as a result of extensive bleaching, which has led to large-scale mortality of coral. That coral then becomes brittle when it dies, and the three-dimensional structure that the reef has been built by the corals becomes very two-dimensional and very flat. Or in the case of many sites around Southeast Asia, as Trias mentioned, blast fishing has become a major, or is a major problem to reef systems in this part of the world. And the fact that this has become increasingly more common meant that, looking back almost sort of 40 years ago, there was a huge rise in reef restoration to start rebuilding habitats like this. And that restoration initially started focusing using asexual fragmentation, and there's many examples of this in places around the world. And this is a really important tool of building the structural complexity that's needed to put back function into these ecosystems. Through increasing that structural complexity, the main driving force of that is kickstarting ecosystem recovery and essentially rebuilding fisheries in these important areas. So we have examples of the work that Mars and my colleagues are doing here in Indonesia, but also locations across the Caribbean. Now, like I say, this is a really important tool. It's used to build that structural complexity. Some of the challenges of only using asexual fragmentation, though, is we know that our oceans are warming and these stress events are becoming increasingly more common. And the challenge with only using asexual fragmentation is we may be limiting the genetic variation or genetic diversity of our outplanted coral colonies. And if the susceptibility of the genotype, so the fragments that have come from the donor colony, if that susceptibility is high from the donor colony, then obviously the same response is seen in the juvenile or the fragments that are planted out. And so this sort of situation of seeing increased bleaching has meant that a lot of people have been focusing on sexual reproduction as a tool in the restoration toolbox, mainly to try and focus on increasing the genetic diversity of the corals that are planted out, whether we can use the very productive, predominantly annual reproductive events to collect many hundreds of thousands, potentially millions of gametes, and can we use that to increase the scale at which restoration can be delivered at? And then taking that a step further, using very novel and cutting-edge techniques to try and increase the genetic diversity and the resilience of the populations that are produced during these reproductive events. So here we have a number of examples, Peter Harrison's work of large-scale collection of gametes from coral spawning slicks.
Jadi di sini kita juga melihat koleksi dari pengumpulan dari gamit slick.
Onto settlement plugs and distributing those seeding units. And then the works
di sini juga kita mendistribusi
Institute for Marine Science or colleagues at Newcastle University in the UK that are really advancing these approaches of human-assisted evolution, of trying to create stronger next generations of corals to withstand some of these impacts of climate change. And this focus on increased genetic diversity through sexual reproduction has really fascinated me, and it's really the reason why I've focused on reproduction for the past 15 to 16 years. So when we think about reproduction, we can broadly split corals into two reproductive modes. We have the brooding species. About 15% of hard corals are brooders, and those are characterized by internal fertilization, where the embryos are developing inside the coral themselves, and they're then releasing fully formed plannular larvae out into the water. Those plannular larvae often will already be inoculated with the zooxanthellae, and these plannulation events generally produce fewer larvae than the broadcast spawning corals, but they aren't necessarily just linked to a single reproductive output throughout the year. Often these plannulation events are linked to the lunar cycle, and so you will have a peak of plannulation potentially hitting new moon or full moon, it depends on the species. That will then drop down as we get either to new moon or full moon, depending on the species, and then pick back up again the following month. And that can happen over many months. So while they're producing smaller number of larvae, they can produce them over a longer period of time. Generally, the settlement of brooders happens more locally to the parents. So the genetic connectivity is often quite close in brooding populations. What complicates the sexual reproduction quite a bit is the fact that brooders can also asexually planulate and produce asexual clones in planular form. So it's quite a complex story with brooders. But then we have the majority of corals, about 85% of those are broadcast spawners of the hard coral species. And those can then be split further down into gonagoristic species. So colonies can either be male or female or hermaphrodites. And hermaphroditic broadcast spawning is the most common reproductive mode for Scleractinia corals. So hermaphrodites more commonly are producing both eggs and sperm within the same colony. So when we think about Acropora are all hermaphroditic broadcast spawners producing egg-sperm packages with both eggs and sperm in. But there are species that can be sequential hermaphrodites. They can be females one year, they can alternate to males another year, and so on and so forth. The characteristics of all of these broadcast spawners, though, is fertilization is occurring externally. So the embryos are developing in the water column, often floating at the surface, where they'll then form a larvae that will settle, form the primary polyp, and then grow into the coral colony. And more research has been done around the world on this. The research started on the Great Barrier Reef, looking at the synchronous mass spawning that's been well documented in the early 80s in the Great Barrier Reef. But other sites can also be asynchronous, where there's different species spawning at different times of the year. So rather than seeing this mass spawning, you can have a more protracted spawning type of pattern. So just thinking about the broadcast coral spawning life cycle, we have an adult colony that releases an egg sperm package in the example of a hermaphrodite. That package breaks apart, fertilization then is happening up at the water surface. We then have the cells dividing over, depending on the species, anywhere between 2 to 4 days. This embryological development happens, forms the planular larvae, and at this stage it's dropping out of the water to find its settlement location on the reef. That then forms the primary polyp, which then divides into the colony, and we complete our life cycle like this. So when I set myself the goal back in 2012, to try and figure out how to induce broadcast spawning in aquariums back in London. There have been spawning events happening in aquariums, but it had always been accidental. And so I wanted to try and understand what are the environmental factors that drive reproduction in the wild, and whether I could then replicate those conditions in an aquarium setting and then have predictable spawning back in London in the UK. And if that could be achieved, could you then have access to these egg sperm, these gametes, a long way from the reef, which would then open up a new area of research to be able to study reproduction, not necessarily associated with the reef location. And so in my initial sort of reading up, the thought process is that spawning is driven by progressively finer scales of environmental parameters that ultimately cue the coral up into releasing these egg sperm packages. When I initially started looking at this, the thought was a colony had to hit a certain size. This was the thinking back about 15 years ago. I think we now know that it's potentially a combination of both size and the age of the polyp. What's also important is the corals need to be feeding. Heterotrophy plays a really significant role in gamete production. And so once you have a colony at the right age and size, food acquisition through heterotrophy is probably a very important factor. And then as we move through the seasonal change in temperature, that triggers the coral to produce the eggs and sperm. And then as we move through the year, we then the coral starts queuing up to the photoperiod. and also the radiation curve of the Earth going around the sun. And then finally, the final trigger appears to be the number of nights after full moon. And so the current thinking is with the length of the day will set them, or the temperature and the length of the day will set the month, and then the night after full moon is triggered by the lunar cycle. So as the sun sets, the period of absolute darkness after sunset to moon rising extends by about 48 minutes a day. And as the period of absolute darkness increases, there's regulation of genes inside the coral, which ultimately triggers the coral to release their gametes. So taking all of these parameters into consideration, I started designing spawning systems back in London that could replicate all of these environmental parameters. And so the early stages were quite rudimentary. Behind this big blackout tent is a series of aquariums. And by each, or on each of these aquariums was a microprocessor, and I could program things like the temperature change over the season, the sunlight dynamics, so the photoperiod and the solar radiation curves, and then also the lunar cycles could all be replicated with LED lighting and a computer microprocessor. And so that initial goal was, could we get predictable spawning of a cropper to start with? And it resulted in the world's first predictable broadcast spawning within 8 months of starting this program. And at that point, we knew we were really onto something. The fact that we had the cameras set, we knew the night that the spawning was going to happen, it really created a platform to allow a whole range of different research to take place. And we've improved the system design over time and refined it as more sort of technology in the aquarium industry became available. We could actually put more sophistication into the systems and improve them as time went on. So I'm going to touch on a few examples of different research that we've been able to do at the museum in collaboration with different partners, and really writing up this methodology was part of my the focus of my PhD. So the first, or one of the first papers I published was the actual system design. So how do we go about actually building the system, the type of computer code that we use to replicate all of the seasonality that we could put into these systems, and also things like the husbandry and the water chemistry management to try and replicate a natural reef as closely as possible. That was the platform that, like I say, allowed a significant amount of work to take place over the past 13 years or so. And the way I think about it is, land-based spawning is the broader umbrella, so creating that platform. But then underneath that sits a number of interesting areas that we can study reproduction. So whether that is fundamentals of reproduction, there's still so many species we don't know how they spawn, when they spawn. And the beauty of being able to do this in an aquarium is you can be looking in that aquarium 365 days a year. And it really gives you an incredible window to understand reproductive behavior of corals really, really nicely and discover new insights. We, and not only that, things like parameters around hybridization, how does that then lead on to hybridization vigor? Is there, reproduction in cold water corals or tropical corals? It opens up a lot of opportunity. These reproductive events are very productive, but they're also, corals are very vulnerable in the early stages of settlement. So how do we use husbandry techniques to start increasing that productivity so we can produce more corals from these spawning events? And this led me down a route of thinking about co-culturing, so rearing corals alongside different herbivores to control algal biomass and increase productivity. And then unique power of ex situ spawning. I'm going to touch a little bit on this. How can we manipulate spawning at various temporal scales to create more access to spawning? And then how do we apply this to to reef restoration approaches. And then sadly, as a result of declining populations of species, how do we use land-based spawning techniques, both as living biobanks for endangered species, but how can we increase the number and the population of those endangered species? And sort of in combined over the years, we've spawned now over 60 species of corals, predominantly acroporas, but we've been working and broadening the range of the different genuses that we've been working on over the last sort of 5-6 years, really. So just unlocking some of the reproductive unknowns, I sort of use this as one example. This is in the aquarium trade, we call this a large polyp stony coral, Homophilia australis. It's an endemic species to predominantly Eastern Australia. It's a very valuable coral for the aquarium trade. And as a result of that, it's a single polyp. It doesn't sort of lend itself very well to asexual fragmentation. And so we really need to think about how can we sexually reproduce these types of corals so we can create a much more sustainable way of creating income for reef communities. And here in Indonesia, there's many species similar to this, things like Trachophilia jeffreyi, Catulophilia, Jardinii. All of these are very much in the same area or same sort of consideration as these types of species. The thing is, nothing was known about how this particular species reproduced. And so this was a study I ran a few years ago to really unlock the secrets of this species, to then create a platform of how we could start thinking about aquaculture of this in a more sustainable way. And, I wanted to document the embryological stages of development of this species, but then also learn something about how quickly can this grow and become a viable coral size to sell into the industry. Previously, it was thought that collecting a coral like this, you may be collecting a 20-year-old coral, and because the supply for the aquarium industry is solely driven by wild harvest, this is potentially a very vulnerable species for overcollection. And so, through sexual reproduction, can we reduce that fishing pressure, really? So it's just one example of unlocking some of the reproductive unknowns of species, and there's a number of different examples of this. We've then gone on to sort of use this knowledge of reproduction with a big project with Oxford University and London Zoo, where there's a critically endangered coral from the Indian Ocean, and we're given permits to collect a number of these to bring back to the museum to act as a living biobank and protect the species from going extinct. It's really gone through a bottleneck in its population, and there are less than 200 individuals that we know of left in the wild. And so by bringing this biobank back, part of the process of bringing these corals to the UK, partly this was making sure that we didn't lose the species, but also how can we figure out its reproduction, which we've now done. Again, nothing was known about the reproduction in this region, and we've understood now it's a brooding species, and we now have just over 100 juveniles now growing out from those 12 individuals. So just sort of highlights the type of work that now can be done through these land-based living biobanks. And colleagues in Florida are working on exactly the same type of work of this, but for Caribbean species to create these biobanks. And once you spawn from that brood stock, can you then start reintroducing the juveniles that are produced? There's other examples of often we may have a spawning where we only have a single individual of a species spawning, and it creates opportunities to understand where the something called pre-zygotic barriers are between hybridizing of particularly around acropora species. Where you're getting these mass spawning events in places like the Great Barrier Reef. Obviously, many species are spawning at the same time, and hybrids can sometimes be produced through these reproductive events. And there's some indication that this hybridization vigor, one, it may be part of an evolutionary stepping stone or part of evolution of creating new species. but also through hybridization vigor, you create stronger offspring for restoration. So this example here was one of those types of hybridizations that we trialed at the museum. We've probably looked at maybe 80 different crosses between species within Acropora. Most of them don't work, but some do. So this example here, where I've crossed Meliopora with Meliopora, we take a photograph of the eggs after fertilization, about 2 hours after, and simply counting a ratio between ones that are dividing and ones that haven't divided gives you your percentage fertilization success. In the example down here, we've taken eggs from Meliopora and sperm from Acropora grandis. And while the fertilization is quite low here, only 17%, it is able to hybridize, or they are able to hybridize between the eggs and the sperm. When you're working with colonies like this, or fertilization batches like this, you have to work very, very hard to rear these juveniles, because the low fertilization means that the ones that haven't fertilized will start polluting the culture. And so you have to clean up the culture a lot over the 4 days to get successful settlement and be able to study this in more detail. When you're doing this type of work, it's really important to look at self-fertilization blanks to make sure that the fertilization from the pairwise cross is true. So what's happened here is I've taken egg sperm bundles from the grandis, just left those, and they can't self-fertilize. And so you should be seeing 0 fertilization in your self-fertilization blank. But it gives you an example of the type of work that you can then start thinking about. We stumble across this by accident, where we shifted spawning from Singapore hyacinthus with Australia. So hyacinthus normally spawns in March and April in Singapore. What I did is I phase shifted them to overlap with Australian corals. And this gave us an opportunity to start studying both intra and interspecific hybridization between different geographical zones. And the idea behind this is Singapore has naturally much warmer water than Australia. And so within the genetic code of the corals that I transported from Australia, these can naturally handle warmer waters. And so we ran a study where we created pairwise crosses from Singapore and Australia. So we made an Australia-Australia cross, a Singapore-Australia cross, and an Australia-Singapore cross. And then ran a thermal stress assay of both fragments from the parents and also the juveniles. Those juveniles and the adults we then snap froze in liquid nitrogen and looked at the metabolomic profiles of both the adults and the juveniles. And importantly, what we found is the juveniles are really very flexible when they're in their early stages of life. And so if you create a stress event for juvenile corals, they can adapt much, much better than adults. And so this gives us one indication that if we're thinking about using land-based spawning for restoration, perhaps it's a good idea post-settlement to provide some stress, controlled stress, at that early age to pre-adapt them before you plant them out into an ocean, and hopefully provide more resilience as time goes on. We took this then a step further to then think about, well, how can we phase shift to create more spawning events in the year? And so we used 3 different research systems focusing on Galaxia fascicularis. We had one that was in sync with Australia. We then phase shifted by 4 months another system to create a 4-month out of season spawn, and then we did the same with an 8-month shifted system. And so rather than having just the normal spawning event, by phase shifting we've increased the access to eggs and sperm, and can that then create a nice platform to further study reproduction, but also can you produce more reproductive output for restoration? This was the platform that we were thinking about. And this work then led on to really the formation of what was originally called the Coral Spawning Lab, and it's now rebranded to Coral Spawning International. But the first project we got asked to do was build a system for KAUST University. And what the goal was is they wanted 24 spawning events a year. So we designed systems that would have ultimately 12 components to it. And by focusing on 2 species that naturally spawn 1 month apart, could we create 24 spawning events? And really to create as much access for CRISPR-Cas9 gene editing work. But it went way further than we actually realized or thought was potentially possible. In the first year, we had 95 spawning events, and that yielded enough material for 30 different attempts at CRISPR-Cas9. So all of a sudden, we're significantly increasing that access and the type of research that can be done. So it's a very powerful tool. That then ultimately led on to starting a company, building coral spawning systems and sending them around the world to support other research groups and restoration practitioners to produce material for restoration. And so we sent about 80 systems to 15 countries around the world. About half of those are used for pure research to understand reproduction at a better level, whether that be around CRISPR-Cas9 or studies or probiotics, whatever the
kami
research.
lakukan untuk penelitian, untuk studi, untuk
restoration. And so, the more we send this out, the more there's a tool to support different outcomes, really. And then thinking about, my work now with the Mars Sustainable Solutions team. My expertise is in coral reproduction, understanding coral reproduction, but from an ex situ setup. And so I've joined the Mars team part-time to try and use my knowledge and provide some assistance to predominantly the Indonesian team. But our goal is to focus on trialing in situ coral spawning to increase the scale at which we can deliver this restoration, with a long-term view that this will be rolled out at the other partner sites that Mars are working with. And so the goals are quite challenging because we want to do this potentially in a slightly different way. The traditional way of working with coral spawning in situ is you put these sort of small-scale nets over individual colonies. You need to have divers in the water. And a buddy pair of diver will sit with one individual coral, or you could be putting a couple of these, but it's quite diver intensive and you have to do night diving. Out in Bontasua, there's not the option of diving at night, so we need to think about things slightly differently. We want to reduce the cost of this as a result, and we don't want to be doing sort of individual night dives. We want to semi-automate the process and also make an approach that can be delivered at a community-led restoration, with the goals of increasing that genetic diversity within the restoration toolbox, and then start thinking about how do we then implement more selective breeding approaches where we can know something about potentially the thermal resilience of the genotypes that we're working on, so we can increase the resilience of the juveniles that are produced. And then the challenges of how do we deliver that at scale, and then really thinking about the broader scale ecological impact of our restoration. So we have the physical footprint of our restoration, and once that physical footprint of the restoration the colonies are healthy and they're reproducing, how far does that broader scale ecological impact have, extending far beyond the restoration, and therefore give us some answer about the scalability of the physical restoration itself. And so this is early stages. We have within the team, the Indonesian team here at Mars, we've been putting in a program to understand first the reproductive behavior that's going on at Hope Reef off Bontasua. And really this first process is you need to understand how the species are reproducing, what time of year they're reproducing, and there's some simple techniques, but they need to be done in a targeted way. The first is we look at when the full moon is, and a few days before the full moon, we're very carefully removing branches from acroporas. At the moment, we're focusing on acroporas, and we're looking at the egg development stages inside the cross section of the coral. And so when the eggs are small and white, they still have a number of months to go. They then become larger and white. And when we see this pigmentation coming into the oocytes, the egg themselves, we know that individual colony is going to spawn at some point around the full moon, potentially. There are species that go in new moon, but the majority of them are going around the full moon. And so when we see that pigmented eggs, the next stage of the process, particularly if we're not diving at night, is put simple spawn collectors over those individual colonies. And as they release the eggs, they're then caught into this bottle, and you can then look at this the following morning and understand that coral released its egg sperm packages the night before. And so in this way, we can do it without night diving. It doesn't tell us what time of day or time of night the spawnings happen, only the number of nights after the full moon. And so colleagues here, my Indonesian colleagues have been working on this every single month since February this year. And we've learned an awful lot about the reproductive behavior that's going on here in Bonsuur. And so Agung, who's done all of this work, is just doing a brilliant job, an absolutely amazing job. So every single month he's been sampling the corals, taking cross-section images, and we have got close to 30 species that we now know the month at which the spawning's taking place. And so this is the first year of creating the platform that we can then build on from next year onwards. So we see these white eggs. This individual's not ready, but as soon as we see that pigmented egg, that's now spawning within the month that this sampling has been taking place on. The majority of the sampling effort has been focused on Acropora, but we have been looking at a few other species, Merlinids and Hyenophora as well. And then those gamete collection cups are put, or collection devices are put on at the point of sampling of seeing the pigmented eggs. And then Agung has been coming back, taking these images, and we see the broken down egg sperm bundles. So the purpose of this is not to rear the egg sperm at this stage. It's simply to define the month at which that species is spawning. And so if you come down in the morning and you see this milky and these eggs are sort of broken down, we know that obviously that individual then spawned the night before. And by documenting that, we're starting to build a picture of which species is going which month and the number of night after full moon. And now, this is really similar to the type of work that I was engaged with in the Maldives over the last few years. When we look at the Reef Rehabilitation Manual, for example, the Maldives, it was sort of said that the February-March spawning window was the peak spawning. There probably still is a peak spawn. We ran a very similar program in the Maldives where we sampled different species every single month for the year. We found a really, really spread out spawning season, not just 2 months, but 6 months of spawning. And the same pattern we're starting to understand is happening in Indonesia, which is really amazing because that gives you, rather than thinking of spawning just happening in November, December, which is the sort of general pattern in the Great Barrier Reef, if you have spawning spread out through the year, you can really start thinking about a conveyor belt of production. So 1 month you'll be working with a certain species, the following month you could be working with another species. And so can we start increasing the production efficiency of dealing with these spawning events rather than having this small window of time, a very spread out window of time. And the more we learn about the species here in Indonesia, my gut feel is we're going to see spawning probably every month of the year, and we'll start seeing spawning not just around full moon. There are already known to be species that spawn at new moon. We'll start filling in more and more of the gaps because there's so many species that are there to be discovered and understood their reproductive behavior. And so our plans moving forward, we have taken inspiration from this paper from Suzuki in Japan, that looked at this large-scale collection of gametes in a semi-automated way. So here, the colleagues, we put our first trial of a coral incubator in April this year. So we make a cluster of 7 reef stars underneath, and then you put 7 genotypes of the species that we want to focus on. We then have a large-scale gamete collection comb, which takes the gametes up to a surface ring and a surface incubator, so that fertilization, embryological rearing, and larval development could all happen in a semi-automated way. So by putting this device on a few days before spawning, you can then stand back and leave it and just keep coming back each morning to see if they've spawned, and then allow that settlement to take place. And there's a choice. Once you have the larvae, you can choose what you want to do with it, whether it's direct larval seeding onto the reef, or whether you want to do targeted seeding onto settlement substrates. There's lots and lots of choice that can be made. We learned a lot about this initial trial in April. I'm here in Makassar now. These last 2 weeks we've been trialing modified versions, which we called our Mark II and Mark III version, ready to start implementing sort of large-scale collection of this from next year onwards. So lots of work that's going on, a lot of very exciting work. I think there's huge potential in this. And thank you very much for listening. Thank you for inviting me to speak and talk to you today. And I'm very happy to take any questions. And there's some information on the various websites here if you'd like to learn more about the different areas of work from Mars, from the Horniman Museum and BioCSL in the spawning systems that we manufacture. Thank you.